Hello, I'm Nicole Gatandia, and I will be presenting my thesis on the effects of dietary intake on the gut microbiome and depression, which I could not have done without the help of Dr. Learmonth. The first 2 decades of the 20th century marked the beginning stages of probiotic treatments, as strains of bacteria were sold and advertised as a means to improve mental health. During the 1920s, however, this practice ended due to the lack of science that would support the link between gut microbes and psychiatric well-being. A renewed interest in the subject sought to connect the functions of the gut microbiome to neurodevelopment and depressive disorders. Researchers have labeled the trillions of microorganisms residing in the gastrointestinal tract as the gut microbiome. It is composed of bacteria, archaea, fungi, and viruses. Microbial richness and diversity marks a healthy gut microbiome. Meanwhile, an unhealthy gut microbiome is composed of pathogenic bacteria or disease-causing bacteria, which induces inflammation. Several external factors influence its makeup, including age, genetics, health, antibiotic use, diet, and stress. This is important because the state of the gut microbiome influences the brain. This is possible because of the gut brain axis. The gut brain axis allows for the bidirectional communication of the gut and brain, meaning the gut and brain not only communicate but listen to one another. They utilize neural, hormonal, and immunological pathways through the central, autonomic, and enteric nervous system. The extensive impact of the gut brain axis and its communication through various pathways allow it to have an enormous influence on the host's homeostasis. When a shift in the gut microbiome occurs, different functions in the body react. As I mentioned prior, stress and diet influence the composition of the gut microbiome. Through the autonomic nervous system, stress signals are transmitted to the gut. Heightened inflammation induced by stress triggers pathogenic bacteria, which reduces the number of beneficial lactic acid bacteria present in a normal gut. In one study, fecal sampling allowed researchers to observe the bacterial levels of college students decreased within days as they documented a high-stress exam week. On the first day of exams, microbiota makeup was similar to that of low-stress weeks. However, by the second day of the exam week, the bacterial levels had significantly dropped. The normal state of the gut microbiome continued to be significantly different until day 5, when testing had ceased. Diet is also one of the leading factors in altering adult microbiome composition. The consumption of probiotics, beneficial living microorganisms, come in the form of supplements or fermented products such as cheese, yogurt, and pickles. Prebiotics are not fully digested, but offer nourishment to microorganisms pre-existing in the gut. The diversity and richness in the microbiome of Americans is 1/3 lower than rural countries because the westernized diet is characterized by foods high in animal protein, fats, and artificial additives, which lack foods high in fiber, vitamins, and minerals. Fiber, which is highly consumed in plant-based diets, allow the microbiome to flourish. Due to the many connections the gut-brain axis is involved in, it is likely that psychological stress and dysfunction in one of the many pathways may result in major depression. An individual's metabolism and their immune, endocrine, and nervous system are all linked in the gut-brain axis, which explains why depressed patients may experience abnormalities in their metabolic rate and appetite. Clinical studies have demonstrated that patients with depression have different composition of gut microbiota than their healthy counterparts. Despite the lack of distinction between the various types of microbiota between the groups, it is evident that depressed patients have significantly less diverse and rich microbiota than the control. In another study, transferring the fecal microbiota of depressed patients into germ-free mice resulted in mice presenting depressive symptoms, such as anhedonia, the inability to experience pleasure. Probiotics, live and helpful microorganisms, alleviated depressive and anxiety-like behavior for the rodents. Treated groups illustrated less symptoms in the failed swim test, which monitors the time mice spend swimming or passively floating. Since mice are considered natural swimmers, mice who remain immobile are seen as expressing depressive-like symptoms. Probiotic-treated mice spent more time mobile during the failed swim test compared to the control group. Using a dietary intake, stress, and depression measure, this study examines the effects of diet and stress on depression. I hypothesize that the consumption of foods that support gut microbiome growth and diversity will correlate with lower depression scores. As well, stress scores are also predicted to correlate with depression scores. I gathered participants through an online survey. They gave consent and disclosed their gender. First, they were asked to document their dietary intake. A chart with 20 foods was presented. Each participant then answered how many times they consumed each food from 5 to 7, 3 to 4, 1 to 2, or 0 times per week. To measure stress, participants completed the undergraduate sources of stress questionnaire, and for depression, the Beck Depression Inventory 2. This is what participants saw when documenting their dietary intake. The first 10 foods are foods that other research has deemed healthy for the gut microbiome. Bananas are prebiotics, cheese and yogurt are probiotics, fruits, vegetables, and lean protein, such as chicken, correlates with a lower risk of depression, and nuts protect against gut inflammation. Meanwhile, the last 10 foods have been shown to disrupt the gut microbiome makeup or cause inflammation in the gut. I had 22 male participants and 65 female participants. Overall, the correlation between dietary intake and depression was not significant. However, when gender was analyzed separately, the correlation was significant. This is a visual representation of female food scores going up while their depression scores are going down. Results show that dietary intake is more likely to affect depression in females, which is interesting because a majority of the articles I read did not analyze gender separately. Therefore, future studies should document and analyze gender to see if there is a difference between males and females. Although my study was non-invasive, if the resources allow, future studies should examine fecal matter to identify bacterial richness and diversity, as well as the specific bacterial strains involved. In the future, it is expected that therapies targeting the gut microbiome will play an important role in the treatment and prevention of depression. Thank you so much for listening to my presentation. I would like to thank Dr. Liermont, as she not only guided me through this whole process, but inspired this project. Thank you.